Hello once again. In Chapter 3, we are going to look at cell structure and function of both the prokaryotic cell and the eukaryotic cell, which you are probably more familiar with. All living organisms possess certain characteristics of life. Responsiveness, this is both to the environment as well as internal changes. Metabolism for energy production, this includes ingestion of food, digestion of the food, absorption, circulation, respiration, and excretion of waste products. All living organisms must be able to grow, and all living organisms must be able to reproduce on their own. There are some examples of types of cells. For example, the prokaryote, which is the bacteria here. The others are eukaryotic cells because they did or do have a nucleus, for example, the neutrophil, or the human red blood cell, which as a mature cell has no nucleus, but did contain one in its early development, our good old friend the paramecium, and algae. Prokaryote comes from the Greek words meaning pre or before nucleus. We define prokaryotic cells today as those that have no membrane-bound nucleus and no membrane-bound organelles. They do contain genetic material, it's just not contained within a nuclear membrane. Whereas eukaryote comes from the Greek words meaning true or good nucleus. So therefore, eukaryotic cells have a membrane-bound nucleus that contains the genetic code of the cell as well as the membrane-bound organelles such as mitochondria, Golgi apparatus, endoplasmic reticulum, etc. We can compare the prokaryote and the eukaryotic cell. In the prokaryotic cell, the chromosome, or the genetic material, there is one circular chromosome not contained within a membrane, whereas in the eukaryotic cell, there are paired chromosomes, linear chromosomes actually, that are contained within a nuclear membrane. The prokaryotic cell has no histone proteins associated with its DNA, while eukaryotic cells do have these histone proteins. Prokaryotic cells have no membrane-bound organelles, while the eukaryotic cell does. In the prokaryotic cell, if it's a bacteria, there is peptidoglycan in the cell wall. If it's in the kingdom Archaea, it's pseudomurin, which is another protein found in the cell walls. In eukaryotic cells that have cell membranes, it's usually made up of a polysaccharide sugar structure that makes up the cell walls, such as cellulose in plant cell walls, chitin in fungal cell walls. Prokaryotic cells tend to be much smaller overall than eukaryotic cells, with most of them averaging about 1 micron in diameter, whereas eukaryotic cells can have a much larger range, anywhere from 10 to about 140 or so microns. The prokaryotic cells primarily reproduce by binary fission. This simply means they make a copy of their one singular circular chromosome, split in half, with each of the two daughter cells now containing a copy of the circular chromosome, whereas the eukaryotic cells create the mitotic spindle in mitosis. This figure of a typical prokaryotic cell is found on page 57 of your textbook. I like this because there's a transmission electron micrograph on one side and the drawing on the other. The nucleoid region, this region here, is where that single circular chromosome is located. You may look at this and say, that's not a circle. Well, the circle is all wadded up to fit the chromosome into the cell. We do have ribosomes, which are organelles, but they are not membrane-bound organelles. We'll have other inclusions and other structures, such as the flagellum, pili, fimbriae, others, cell wall, the glycocalyx. We'll talk about each one of these in detail. This is your typical eukaryotic cell, and you'll notice that the genetic code is contained within the nuclear envelope or nuclear membrane. The nuclear membrane will have nuclear pores that will allow for large structures such as messenger RNA to exit the nucleus. And we have the membrane-bound organelles such as the Golgi apparatus, the endoplasmic reticulum, the mitochondria, the lysosome. Most of these you're very familiar with. Also, eukaryotic cells, because of their size, typically has a very intricate cytoskeleton that gives the three-dimensional shape of the cell. Basic bacterial shapes. The bacillus is a rod-shaped or elongated shape. The coccus is a spherical-shaped bacteria. Bacilli and cocci are the plural form of these two words. Then we have the spiral organisms, and this depends on how twisted you actually are. For example, the vibrio kind of look like a comma. The spirillum may have 3 to 5 or so twists along its long axis, whereas the spirochete is very twisted. Arrangements, how the bacteria are actually arranged within a grouping. So you can have singles, but here we see the pairs. Diplo. What does di mean? Di means two. So diplococci, diplobacilli. Clusters, staphylo is the term for a cluster-like or grape-like cluster. And so we find staphylococci, which you will only find in a coccus formation. You do not have a staphylobacillus. They just don't exist. So when we talk about the arrangement, the cluster is a staphylo. Chains, you can have streptococci and streptobacilli. The prefix strepto means chain-like. Here's an example of Diplococcus. Now you may say this group here looks more like a Staphylococcus, which may be true, but the majority of the arrangements is Diplo. Here you have the Staphylococci, and it's nice that they actually colored these purple so they look more like grapes in a cluster. Here's streptococci, which you can actually see are just like long chains of pearls along a pearl necklace. Other cocci arrangements, we can have the tetrad, where the cocci form in groups of 4, forming a square-shaped cluster, or sarcinae, which is cocci that form in groups of 4 on top of a group of 4, forming basically a cube. Here's a good example of tetrads. Here's the sarcinae. You can tell from some of these arrangements that you can actually see the 8 bacteria that form this cube-like structure. On the bacillus side, we can have the single bacillus, such as this E. coli. We can have diplobacillus, which usually results from binary fission where the cell walls just simply do not completely separate. And you'll notice that most of these are in pairs. However, this one has 1, 2, 3. It's more of a chain, but again we go with majority, and the majority are diplo. This is truly streptobacillus, long chains of these rod-shaped organisms strung together. What about this guy? What do you think? Is he a cocci or is he a bacilli? What's this shape? Some bacteria don't really know. These are all the exact same bacteria. Some of them are more spherical shaped, cocci. Some are more elongated, bacilli. And then some of these mushroom shapes look more like, well, not quite sure what they look like. So we call these guys coccobacillus. Just can't make up their mind what they want to be. Bacillus actually has 2 meanings depending on the way it's used. So I could say the shape of these bacteria is bacillus, but the scientific name of this particular bacteria is Bacillus anthracis. Again, if you look, Bacillus anthracis is italicized, meaning it's the proper name. So I need to understand whether I'm talking about shape or scientific name of the bacteria, depending on how we're using the meaning. We also have some unusually shaped bacteria, such as these star-shaped bacteria. In the Archaea kingdom, many of the organisms will have very odd shapes, similar to this. This is actually a photomicrograph of microbes living in the rumen of a bison, Shown here is a star-shaped aggregate of cells, which we actually call a star shapes, a very thick long rod, numerous cocci, and a few other shorter rods. These organisms in the bison actually aid in digestion of plant material. We can also see rectangular-shaped bacteria and archaea. This actually is an example of a rectangular-shaped archaea. The spiral organisms, here we have the Vibrio. I said Vibrio are kind of comma-shaped. They basically have a half turn along the long axis of the organism, and so therefore they have this kind of comma shape. The spiral or spirillum bacteria, again, have about 3 to 5 twists in the organism. You can see they almost look like a wave form. And then this shows a spirochete, highly twisted, and in fact this is Treponema pallidum, the causative agent of the disease syphilis. We will start looking at the prokaryotic cell from the inside out. On the very external part of most bacterial cells, we have the glycocalyx or the glycocalyces. These typically are a gelatinous sticky substance that surrounds the outside of the cell wall. Typically, they're composed of polysaccharides, polypeptides, or usually actually both. There are two types of glycocalyces that we can find in bacteria. The capsule, which is composed of organized repeating units of organic chemicals, are very neatly organized at the surface and firmly attached to the cell wall. These capsules can be very thick and may prevent the bacteria from being recognized by the host's immune system. I actually call these capsules the invisibility cloak of the bacteria that have them. The slime layer on those that don't have capsule typically is very loosely attached to the cell surface, much less organized than the capsule. Typically will be water soluble because it's mostly going to be sugars, and it provides a sticky layer that allows the prokaryotic cells to attach to surfaces or to each other. Slime layers are very important in something we'll describe later on, known as biofilms. Here's an example of a capsule around some diplococci. And here's a high magnification of a slime layer around this bacterium. The flagella on certain bacteria are responsible for movement. They have long structures that extend beyond the cell surface, and we know these as the flagella. They are not present on all bacteria, and flagella will only be found on bacilli. The structure is composed of a filament, which actually causes the rotational movement, or the propeller-like movement, the hook, which provides the rotation, and the basal body that is actually connected to the cell wall and cell membrane of the bacteria. This basal body anchors the filament and the hook to the cell wall by a rod in a series of either 2 or 4 integral proteins. Here's the drawing. We have the protein rings of the basal body and the rod, the hook, which will provide rotation, and the filament that would continue on up here. This is found in a gram-positive. The gram-positive will have 2 rings in the basal body, and this shows the gram-negative, which has 4 rings in the basal body. Because the gram-negative cell wall is thinner than the gram-positive, we basically need more proteins to actually anchor the flagella to the cell wall and cell membrane. Some books will actually say gram-positive bacteria have 1 ring in the basal body. However, what this means is that we actually have 2 of these proteins within this ring structure. So I actually say that the gram-positive simply has 2 rings. We do know that the flagella is outside the cell wall, the part that moves, and the filament itself is made up of chains of a protein called flagellin. It's attached to a protein hook that provides the rotational movement and anchored to the cell wall and membrane by the basal body. The gram-negative, some books will say, have 2 rings in the basal body, but as you can see here, we actually have 1, 2, 3, 4 rings of protein, so I still refer to this as 4 proteins in the gram negative. Bacterial flagella can be arranged in a number of ways. I've included on this slide how to pronounce these, so I will try to do my best. This arrangement, where you have flagella all around the outside of the bacteria, is known as peritrichous. This one has a single, so mono, and on one pole, and so we call this monotrichous and polar. The liphotrichous is where you have more than one flagella, but they're all located at one pole. And amphitrichous and polar is meaning that they are on both ends of the bacteria. Many of the spiral organisms will show this amphitrichous form of flagella. Here's a good example of a peritrichous flagellum on a bacteria. This happens to be Proteus vulgaris. Do you think this guy can move lots? These are not sperm. These are actually bacteria that have the monotrichous flagellum. It's at one pole, and this makes sense that the bacteria will move opposite of the end of the flagella. It's like having a little motorboat with your motor on the back of the boat. They'll head forward. Even the liphotrichous flagellum, I understand it's like you have more than one motor on the end of the boat. And amphitrichous flagella typically are not used for the actual movement of the bacteria, since most of these are spiral organisms. They will actually move by the endoflagella, we'll talk about in a moment, but these amphitrichous flagella will act as rudders. In these motile cells, they tend to either move towards or away from stimulus, and we call this taxis. I like the name from the Latin because taxis, oh yeah, they move you from point A to point B or away from point A to point B. And so taxis, we can have either chemotaxis, where we are attracted or repelled by a chemical, or phototaxis. What would attract or repel a phototaxic bacteria? Flagellar proteins contain what we refer to as H antigens. This is part of the flagellin protein. These H antigens are useful in distinguishing serovars or variant strains within a species. For example, E. coli 0157H7, the really bad guy of E. coli, that H7 refers to a specific type of flagellin protein. The peritrichous bacteria move by rotating flagella in what's known as the run and tumble. Since you have the motors on all sides of the boat, they have to be able to do movement in a forward direction. And as all of these flagella spin, they typically will begin to tumble or roll around. But at some point, typically, all of the flagella point towards one pole of the bacteria, and they work in coordination and make a forward run. Then they come out of coordination, the bacteria tumbles around, they make another run, then tumble, then run towards the attractant or away from a repellent. The spiral organisms, such as with this spirochete, have an endoflagella. These are sometimes also referred to axial filaments. This is a photomicrograph of the spirochete Leptospira showing this axial filament. Typically, this endoflagella will be anchored at one end, and the rotation will cause the cell to move and also give it its characteristic spiral shape. The endoflagella are located just underneath the cell wall and cell membrane and gives it this corkscrewing action. Here is a video that I'm going to play showing these spirochetes under a microscope. You'll notice that they do tend to be able to follow pretty well in a line, but can move by simply this twisting method. Here's another real good picture of one that is moving. They're hard to keep up with sometimes underneath the microscope. Fimbriae and pili are rod-like proteinaceous extension, but different from the flagella. First of all, neither one of these are really involved with movement. The fimbriae are sticky, bristle-like projections that are used by bacteria to adhere to one another, to host cells, or to substances in the environment. They are typically shorter than flagella, and they serve a very important function in those biofilms we'll discuss later. The pili are tubules composed of a protein called pillin. They are also sometimes referred to as conjugation pili or sex pili. Pilus is the singular of pili. They are longer than fimbriae, but typically shorter than flagella. Most bacteria that form pili have only one or two per cell, and what these pili do is they mediate the transfer of short segments of DNA from one cell to another in what we'll refer to as conjugation. They will have gliding or twitching motions trying to find the other cell in which they want to exchange genetic material. They are not exchanging all of their genetic material, but only pieces and parts, so this is not considered sexual reproduction. Here's a good example of fimbriae on this diplobacilli. You can see that they are much shorter than the flagella, and hopefully from this picture you can get the idea that they actually are sticking these guys down to the environment that they're in. The conjugation pilus will reach out and form what we call the mating bridge between two bacteria, and it is through this pilus that we can then exchange these pieces of DNA. This actually shows one of these pili where it has found a second bacteria. This will slowly draw this bacterium to the other one and form the mating bridge for the exchange of the genetic material. The cell wall of prokaryotic cells is unique. It contains peptidoglycan as the primary protein. Bacterial cell walls provide the structure and the typical shapes of bacteria, and the big thing, it protects cells from internal osmotic forces. Most bacteria have a very high internal osmotic force, meaning that they tend to take up water, and therefore the water would try to rupture the cell membrane. If we did not have the cell wall, the bacterial cells would actually lyse. The primary function of the bacterial cell wall is to prevent osmotic lysis, but they have other functions as well. Some cell walls can assist the cells in attaching to other cells or resisting antimicrobial drugs. We can also target the cell wall of certain bacteria with antibiotics. The bacterial cell wall will give the cells the characteristic shapes, and it's composed of a protein sugar structure known as peptidoglycan. Scientists describe two basic types of bacterial cell walls, the gram-positive and the gram-negative. Let's look at each one of these. Peptidoglycan is a polymer of a disaccharide with two sugars, N-acetylglucosamine and N-acetylmuramic acid, NAG and NAM. You do not have to memorize these structures. Simply know that we have long chains of alternating NAG and NAM that form a rod-like structure. So you have NAG-NAM, 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 etc. These long chains of the sugars are laid down parallel to each other, and just like logs in a raft, need to be tied together. And here we will use a polypeptide cross bridge that will actually then tie these logs together, forming a very strong layer of peptidoglycan. In the gram-positive bacterial cell wall, we have a very relatively thick layer of peptidoglycan. We have numerous layers of these peptidoglycan sheets piled on top of each other. They contain unique polyalcohols called teichoic acids that actually will tie the layers together. It's kind of like in a laminate wood. You have layer upon layer of wood fiber laid on top of each other and glued together. The teichoic acids would be the glue The wood fiber layers would be the peptidoglycan layers. They appear purple following gram staining. And in the acid-fast bacteria, they are basically gram-positives, but also have up to an additional 60% of mycolic acid. The mycolic acid in the acid-fast bacterial cell wall helps the cell survive desiccation or drying out. We also know that the gram-positive cell wall may regulate the movement of cations in and out of this bacterial cell. The polysaccharides found on the surface of the cell wall provide antigenic variation among different species and different serovars within a species. Here's the example from the old text that shows this layer upon layer upon layer peptidoglycan held together by the teichoic acids. So we have a very thick peptidoglycan cell wall. Here's the picture from your book in page 65, and it's kind of like bricks and mortar. And these long strands then of the lipoteichoic acids that help anchor all the brick layers together. Versus gram-negative bacterial cell walls that have only a thin layer peptidoglycan. In fact, it's a single layer of peptidoglycan. What we also have, though, on top of that is a bilayer membrane of phospholipids that will also contain proteins and lipopolysaccharides and other carbohydrates. So in the gram-negative, you have an outer phospholipid membrane, the single layer peptidoglycan, and then the bilayer of phospholipids that form the cell membrane. The outer membrane forms a space between the outer membrane and the cell membrane known as the periplasmic space. Because of the outer bilayer of phospholipids, this may be an impediment to the treatment of disease caused by gram-negative bacteria. And in general, gram-positive bacteria are easier to treat with antimicrobials than are gram-negatives. Gram-negatives will appear pinkish red following the gram staining procedure. This figure also on page 65 shows again that single layer of peptidoglycan, the outer membrane that also has proteins and lipopolysaccharides on its surface, and then the inner cell membrane. In between the two phospholipid membranes, where the single layer peptidoglycan is located, will be the periplasmic space. This also is shown from your old edition of your text, showing that outer membrane, the single layer of peptidoglycan, and the inner cell membrane. Many of the lipopolysaccharides on gram-negatives are designated as O polysaccharides or lipid A. For example, E. coli O157H7, the O157 is a particular strain of polysaccharide found on that gram-negative bacterial cell wall. So, gram-positive cell walls, a very thick multi-layer peptidoglycan, whereas it's a single thin layer peptidoglycan in the gram-negative. You have an outer lipid membrane and the periplasmic space in the gram-negative cell wall. And the gram-positives have the teichoic acids that will help hold the many layers of peptidoglycan together. Gram staining, gram-positives will appear purplish after gram staining, whereas the gram-negatives will appear reddish pink. I always think positive, purple, negative, red. How exactly does the gram stain work? The first stain, the primary stain, crystal violet, will combine with the iodine mordant to form crystal violet iodine crystals within the cell. When gram-positive bacteria undergo the decolorizing wash, the alcohol within the wash dehydrates the peptidoglycan, forming a complete sealed barrier. Therefore, the crystal violet iodine crystals cannot leave the cells, and the cell will retain the crystal violet or purple color. Whereas gram-negatives, the alcohol actually dissolves the outer phospholipid membrane because it's a fat and will punch holes in that thin layer peptidoglycan. Therefore, the decolorizer will wash the crystal violet iodine crystals out of the gram-negative, leaving them colorless. Then when we put the secondary safranin stain on, the gram-negative take up that reddish-colored stain and appear reddish-pink. Atypical cell walls, we've talked about the acid-fast cell walls that are basically gram-positives, but have the addition of mycolic acid. This mycolic acid is bound to the peptidoglycan layer. There are two major genuses of bacteria that are acid-fast that cause disease in humans, the Mycobacterium species and the Nocardia species. In an acid-fast stain, the acid-fast bacteria will appear red. Here is Mycobacterium tuberculosis, which is an acid-fast bacteria, and the non-acid-fast bacteria that take up the methylene blue secondary stain of the acid-fast staining will appear this bluish color. So you have to know what is the stain that you are looking at. Mycoplasmas are the smallest of the bacteria, and they are basically the only bacteria that lack a cell wall. They are very small, averaging about 0.5 microns in diameter, and they have many, many sterols in their plasma membrane. This is basically large chunks of cholesterol scattered throughout the plasma membrane, which gives enough strength for the mycoplasmas not to undergo osmotic lysis. So therefore, they do not need the cell wall. Whether or not they had one at one time and decided to lose it is unknown. The archaea domain are either wall-less, or when they do have cell walls, it is made up of the protein called pseudomurine, which lacks the NAM and the D amino acids found in peptidoglycan. Here's a close-up of Mycoplasma bacteria. This is not a cell wall, but actually the cell membrane under this high magnification. This is about 0.5 microns in diameter. This is one of the archaea, one of the first ones actually we discovered, Staphylodermus marianus. Let's break the words down. Staphylo, so they occur in clusters, even though we only see a single one here. Thermus, meaning they must like it hot. And Marianus, deep ocean. This is an extremophile living in the deep ocean thermal vents. It's both a thermophile in very hot temperatures as well as a barophile living under very high pressure. This was first discovered in the Marianas Trench in the Pacific Ocean at 7 miles down. We can damage the cell wall and actually kill the bacteria. Let me ask this question. You get a paper cut. Everybody's had one. And what's one of the first things that you will do after you say the words? Usually, you stick it in your mouth. Why? Well, we'll talk a little bit about this in class. But in saliva and other body fluids, we have enzymes called lysozymes that will digest the peptidoglycan in the cell wall. In gram-positives, the lysozymes can attack directly, while in the gram-negatives, they're not as effective because of the outer phospholipid membrane. But think about it. Staphylococcus, streptococcus, the common bacteria that would be found in a paper cut, are both gram-positives, and so those lysozymes actually begin an attack on the bacteria at the very beginning. So here's the question. Should you lick your surgical wounds? Think about it. Penicillin class drugs inhibit peptide bridges in the peptidoglycan layer, therefore punch holes in the cell wall, especially of gram-positives, and penicillin is very effective with gram-positives in general. When the cell wall is damaged for whatever reason, the cells begin to expand and form what we call L forms, which are wall-less cells that swell into irregular shapes because water is entering the cell. Protoplasts and spheroplasts are basically the same thing. The protoplast is a wall-less cell where the wall is either completely or partially removed by using either mechanical or enzymatic means, and a spheroplast specifically is a wall-less gram-positive cell. Both of these, because the cell wall is damaged, are susceptible to osmotic lysis. This shows Proteus mirabilis, which is a bacillus that has undergone an enzymatic attack. What is happening is the cell membrane is swelling because the cell wall is so damaged, and these cells actually are rupturing. This figure shows one of these cells after it has ruptured. Basically, nothing is left. The cell membrane, internal to the cell wall, and it is made up of a bilayer of phospholipids. This is very similar to the cell membrane of eukaryotic cells. The only differences may be the different types of proteins that we see. The phospholipid bilayer will separate the water outside environment from the water inside environment because it's a fat. We'll have peripheral proteins, integral proteins, transmembrane proteins that provide a variety of functions across the cell membrane. Membranes are about as viscous as olive oil. And think about it. Go home, put some olive oil or other oil in water. They separate. This is what gives the membrane its selective permeability. Proteins can move within the layer of phospholipids to function at various places. So I may take a snapshot of this cell membrane today, and it would look like this. Tomorrow, I might take the same snapshot of the same place and find either more or less proteins depending on the function today. The phospholipids can rotate and move laterally within their side, but cannot flip to the other side because the phospholipid fatty acid chains will orient towards each other, with the polar heads being oriented towards the water environments, both outside and inside the cell. The functions of the cytoplasmic membrane: they provide energy storage and enzymes for ATP production within the prokaryotic cells. They can harvest light energy in photosynthetic bacteria on foldings of the membrane called the chromatophores or thylakoids. They are selectively permeable. They are naturally impermeable to most substances due to the fact that the phospholipid bilayer is a fat and most substances are water soluble, but the proteins in the cell wall can allow substances to cross the membrane. This also will maintain the concentration and electrical gradient across the cell wall. Cell membranes can be damaged by alcohols, quaternary ammoniums, which are found in most detergents, and polymyxin antibiotics. All of these will damage the cell membrane and cause leakage of the cell content. This photograph shows the chromatophores on one of our purple bacteria that actually uses photosynthesis for energy production. We said the cell membrane will provide the electrical gradient across the cell membrane, and just like in our cells, the resting membrane potential usually measures around minus 70 millivolts because you have more negative charges inside the cell than outside. Again, these integral proteins, though, can allow for movement of these ions in and out of this cell. Selective permeability allows passages of some molecules while excluding others. Most of these should be reviewed: simple diffusion, facilitated diffusion, osmosis, active transport, endocytosis, be it phagocytosis, which is engulfing particles, or pinocytosis, which is engulfing fluid. The one you're probably not as familiar with is group translocation, where the substance being transported is also being chemically modified at the same time. Simple diffusion is defined as the movement of solute from an area of high concentration of solute to an area of low concentration of solute across a permeable membrane. So, lots here, few here. This is a fat-soluble substance, so it will freely move through the plasma membrane and will simply diffuse through the membrane until we reach equilibrium, equal numbers on both sides. example, if I put a crystal of potassium permanganate in this beaker, where is the high concentration to begin with? What will happen over time? The question is, what will happen as this potassium permanganate dissolves? Will this entire beaker become completely purple and be equal in all areas? Facilitated diffusion is still the movement of a solute from high concentration to low concentration, but now it must go through a transporter protein within the membrane. It cannot just cross anywhere. This is kind of like in the room. We have a door. The only way you get in or out is through the door, not through the walls. So we can have nonspecific transporters that will allow a variety of substance through from high to low, or we can have specific ones that will only bind to certain things and transport them from a high concentration to a low concentration. Osmosis is the movement of water molecules across a selectively permeable membrane from an area of high water concentration to an area of low water concentration. As the water moves from high water to low water, this will create pressure within a cell or organism. This is what we refer to as osmotic pressure. It is defined as the pressure needed to stop the movement of the water across the membrane, or the pressure needed to lift a volume of solution against gravity. So here we have a sugar solution in a dialysis bag attached to a glass tube, but we suspend this dialysis tubing in pure water. To begin with, where is there more water molecules? Inside the bag or outside? What is going to happen as we allow this to sit? In cell membranes of bacteria, we have these transport proteins called aquaporins. Aqua, water. These allow water to move through this fatty lipid membrane, and as it does so, it creates a very high osmotic pressure inside the bacteria. This is why we need that cell wall, to prevent the pressure buildup from rupturing the cell. So here we've come back to our dialysis bag with the sugar solution. What happened? What happened to the level of solution in the glass tube? We also talk about tonicity when we're talking about the principles of osmosis, and this has to do with the pressure. Iso refers to same. So an isotonic solution means that the water is already equal on both sides of the selectively permeable membrane. So if we put a cell in an isotonic solution to it, it means that there's already equal amounts of water inside and outside the cell. Does water move? And the answer is yes, but for every molecule that would move in, one would move out to maintain equilibrium. So nothing happens to the cell. However, if we put a cell into a hypotonic solution, the hypotonic means that there are less solutes, therefore more water, outside this cell. So more water outside, less inside, water moves into the cell. If the cell wall is strong enough, it will contain the swelling against this high osmotic pressure inside. But if the cell wall is weak or damaged, the cell wall will eventually lyse. If we put a normal cell into a hypertonic solution, this means the solution has more solutes, so therefore less water. Water then moves from the high concentration inside the cell to the outside, causing the cytoplasm to shrink. The cell wall does not shrink, but as the cell membrane shrinks or crenates and pulls away from the cell wall, it can actually tear. This will result in plasmolysis, meaning that cell contents now leak out through the damaged cell membrane. Here's a nice normal red blood cell in an isotonic solution. has that nice biconcave look to it. This red blood cell has been put into a hypertonic solution. Water has come out of the cell into the outside, causing the cell to shrink or shrivel. These wrinkles are known as crenation. Here's a red blood cell put in a hypotonic solution. You'll notice it's lost its biconcavity and in fact is about ready to rupture simply because this red blood cell does not have a cell wall to contain the growing pressure inside it. Mechanisms of active transport. Active transport is where we go from low concentration to an area of high concentration. This is not the way something wants to move. Which way does water want to go, up or down the hill? Well, you all should say it should flow down the hill with gravity. But can I take water up a hill? And the answer is yes. I can take a pail, put the water in it, and carry it up the hill. Or since I'm lazy, I'll put a pipe to the top and put an electric motor and pump the water up the hill against gravity. Active transport goes against the concentration gradient, going from low to high. Two other things we'll need. We need a transport protein, and we must use cellular energy. It takes energy for me to walk up that hill or energy to run my electric pump. So we're going to use some ATP to do this. In a uniport system, we are transporting a solute from low concentration to high using a carrier molecule and ATP. We only go in one direction, uni. An antiport is where we're going to have one substance going from low to high and another substance doing the same thing in the opposite direction. Again, we're going to use ATP. Really good example of an antiport system in our bodies is the sodium-potassium pump that will pump sodium out while bringing potassium in. And coupled transport usually has a symport and a uniport together. Symport transport proteins will transport two different molecules at the same time through the protein, but one of those is then transported back out almost immediately through a uniport using ATP. The symport moves the two molecules together in one direction, and we have to have the circle in order to get the square through, but we don't want to keep the circle in, and so we transport it back out against its concentration gradient. Group translocation is where we're going to change the substance as it's being transported. So our transport protein not only is a transport protein, but usually an enzyme as well. Here's an example. Glucose goes from one side of the membrane to the other, but it enters as simple glucose and is changed by the transportation protein into glucose 6-phosphate. This is actually the first step of glycolysis within prokaryotic cells. The cytoplasm is everything inside the cell membrane. This would include the nucleoid region, the DNA, any other pieces of DNA, food granules, and the ribosomes. The nucleoid region is simply the area where the bacterial chromosome is found. This lighter region in this TEM shows the nucleoid region. Remember, I said bacteria have a single circular chromosome, but it gets wadded up here in the nucleoid region. Here's a closer view of an electron micrograph of the nucleoid region. It is this light region. The bacterial ribosomes are found scattered throughout the cytoplasm. These will be used in protein synthesis just as they are in our eukaryotic cells. However, the type of ribosome found in prokaryotes is different from the eukaryotic ribosome. It is known as a 70S ribosome. The ribosomes are made up of two subunits of ribosomal RNA. In this case, we have the small subunit designated as 30S plus the large subunit 50S that will come together on the messenger RNA and form the 70S ribosome. Yes, I know, 50 and 30 does not equal 70. Think of it this way. These two subunits slam together, forming the ribosome, and they get to lose 10 S pounds. So they become a 70S ribosome. I wish that's the way it worked with me. Some bacteria have a very simple helical cytoskeleton. Primarily, these cytoskeletons will be found in the larger spirochetes. This will help give the shape of these elongated bacteria. There are a number of bacterial inclusions that we can find in bacteria. The metachromatic granules, known as Volton, act as phosphate reserves within the bacterial cells. Polysaccharide granules can act as energy reserves. Lipid inclusions can also do this. Sulfur granules as well. The carboxysomes contain ribulose 1,5-diphosphate carboxylates, which is for carbon dioxide fixation. This is found primarily in those photosynthetic bacteria that use CO2 as a carbon source. Gas vacuoles can maintain the buoyancy so that certain bacteria can remain at certain depths within solutions appropriate for their metabolism, such as the archaea methogens must maintain buoyancy to remain within the methane layer of seawater. So they don't want to be too high and they don't want to be too low because otherwise they would die. Some also have magnetosomes. These are protein-covered cylinders filled with iron oxide that are thought to be used by bacterial cells to destroy hydrogen peroxide. This is a picture of some magnetosomes within this bacterium. Interesting thing, since they have iron, they will actually respond to a magnetic field. This bacteria was put in a magnetic field and all the magnetosomes migrated to this area. Endospores are a resting form of bacterial cells that are formed when essential nutrients within the environment are depleted or the environment is no longer conducive for bacterial growth. They are resistant to desiccation, heat, chemicals, freezing temperatures, etc. Certain gram-positive bacteria, such as the Bacillus genus and the Clostridium genus, are endospore formers. When they form the endospore, this process is known as sporulation. If the endospore enters a favorable environment, it will return to the vegetative or living state through the process of germination. Endospores are not considered to be a form of reproduction because you go from one cell to one cell. It is simply a resting state of the cell. Here's a very close-up of an endospore in Bacillus anthracis. This structure in the central part of this cell is the endospore. Eventually, the cell that forms the endospore will die because of lack of nutrients or other things in the environment, and the endospore will remain until the environment changes for the better. In sporulation, we make a copy of the DNA. We put one copy in the cortex. We form a spore coat and an outer spore coat, and eventually the cell that forms the endospore dies, and the endospore remains in the environment. This is an endospore stained slide showing the green spores inside these bacterial cells. You'll notice here, this one basically has no red around it because the cell that formed it has actually already died. The eukaryotic cell. Most of this should be review for you, but we will go through this fairly quickly. Eukaryotic cells also have glycocalyces. They're never as organized as prokaryotic capsules. They're more similar to the slime layers. They help anchor animal cells to each other, and in fact, the cells produce these as part of the tissue matrix. They help strengthen the cell surface. They provide protection against dehydration, and they function in cell-to-cell recognition and communications within tissues and organs. The fungi, algae, plants, and some protozoa have cell walls. Mammalian cells do not. If you do have cell walls, it's composed of a variety of polysaccharides, such as in plant cell walls, it's cellulose. In fungal cell walls, we have cellulose, chitin, and or another sugar known as glucomannan. And algae cell walls are composed of a variety of polysaccharides depending on the type of algae. This shows a cell wall in plants. This shows a fungal cell wall primarily made up of chitin. Certain eukaryotic cells also have flagella and cilia. What is the only human cell that has a flagellum? The flagella and cilia are both composed of protein rods called tubulin. These rod structures are arranged in what we call the 9-2 array. You have 9 doublets around the outer surface and 2 single central microtubules. So, unlike the prokaryotic flagella, we do have the basal body, but the hook basically is not existent. While the prokaryotic flagella moves in a propeller-like motion, the eukaryotic flagella move in an undulating manner. In other words, they whip back and forth. This is a cross-sectional view of the human sperm flagellum. Here you can see the 9 doublets and the 2 small central microtubules. Cilia are shorter and more numerous than flagella, but are also made up of the 9-2 arrangement. They have coordinated beating that propels the cells through their environment. They also can be used on cells to move substances past the surface of the cell, such as the ciliated epithelial cells of our trachea and bronchi. Here's a nice little heart-shaped ciliated organism. And again, in the flagella, we have the whipping motion that will propel the organism in the environment, such as this euglena, and the cilia, which have that coordinated power stroke and recovery phase that will allow for single directional movement. The eukaryotic plasma membrane is the bilayer of phospholipids containing the peripheral proteins, integral proteins, transmembrane proteins. We also have a lot of sterols, such as cholesterol and other steroids that give structural integrity to the plasma membrane of the eukaryotic cell. And then we also will have glycocalyx carbohydrates found on the outer surface. Here's a picture from McGraw-Hill that actually shows a snapshot of the bilayer phospholipids with cholesterol and proteins and the glycolipids found on the external surface. The cytoplasm, again, is everything inside the plasma or cell membrane, but outside the nucleus. The cytosol is the fluid portion of the cytoplasm and is primarily made of water. In eukaryotic cells, because they are larger than prokaryotic cells, we will have cytoskeletons, which are comprised of microfilaments, intermediate filaments, and microtubules. We also will see a circulatory motion that's known as cytoplasmic streaming that will allow movement of the cytoplasm throughout the cell. Here's a stained picture of cheek cells showing the nucleus and the cytoplasm. The cytoskeleton gives the eukaryotic cell its three-dimensional shape, and there are 3 types of fibers that make up the cytoskeleton. The microtubule, made up of the protein tubule, which is about 25 nanometers in diameter. The microfilament, made up of the protein actin, which is about 6 to 7 nanometers in diameter. And an intermediate filament made up of a protein not yet named that's about 10 nanometers in diameter. All of these form an internal skeletal structure of the cell. This picture under fluorescent microscopy shows in the green all of the different fibers making up the cytoskeleton. The ribosomes also involve in protein synthesis in eukaryotic cells, but now we have 2 different types. The primary type of ribosome found in eukaryotic cells is the 80S. This is made up of two subunits, 60S and 40S. Yes, again, I know, they don't add up to 80. Just accept it. They are found on membranes attached to the endoplasmic reticulum or free-floating in the cytoplasm. However, eukaryotic cells also have 70S ribosomes, just like those found in prokaryotic cells. But 70S ribosomes are found only in the chloroplasts, in photosynthetic cells, and mitochondria. They are used to make proteins involved with energy production. Here's a picture of the 40S and 60S subunit forming the 80S ribosome. Organelles found within the cytoplasm include the nucleus, which contains the genetic material of the cell, the endoplasmic reticulum, which is a transport network within the cell, and it's a membrane-bound organelle. The Golgi complex or apparatus, it's a membrane-bound organelle that's involved in membrane formation and secretion, storage of molecules within the cell. The lysosome, which sometimes referred to as the suicide packet of the cell, because it contains digestive enzymes used to break down foreign material, dead and dying organelles. If, however, the lysosomal membrane ruptures, the cell will auto-digest. The digestive enzymes will literally eat the cell from the inside out, killing it. Vacuoles or vesicles that can bring food into the cells, provide support, transportation. The mitochondria, these are the powerhouse of the cells. They are involved in cell respiration and the production of ATP. Chloroplasts also provide ATP in those photosynthetic cells. Peroxisomes, which are membrane-bound organelles that contain enzymes that oxidize fatty acids and destroy hydrogen peroxide. The centrosome, that consists of protein fibers made up of the two centrioles, and the centrosome is involved in mitosis, cellular reproduction. Here you have the nucleus. In this cell, the nuclear membrane or envelope contains the nucleolus that is involved in RNA production and chromatin. This is DNA in its relaxed form where we are able to read it. If this cell were reproducing, the chromatin would tightly coil up into the chromosomes and be visible. The endoplasmic reticulum actually comes in two forms, the rough that has associated ribosomes on its outer surface and the smooth. Both forms of the endoplasmic reticulum are involved in transportation of substances within the cytoplasm, packaging, and even release of the product to the outside of the cell. The Golgi body or apparatus is a membrane-bound organelle made up of 6 to 8 flattened packets of membrane. The Golgi body is involved in secretion, making secretory vesicles or transfer vesicles, storage within the cistern structures, and also modification of many substances. Here's a close-up of Golgi apparatus. The lysosomes, peroxisomes, vesicles, and vacuoles, you cannot actually tell the difference. It's simply what's inside of them that makes them a peroxisome versus a lysosome. I would not expect you to be able to tell me that this was a peroxisome or a lysosome, because you can't really tell until you analyze what's inside the membrane. Here's a central vacuole. This would be something like found in an adipose cell, and the central vacuole would contain fat. Vesicles play a role in the destruction of a phagocytized pathogen. This bacterium is undergoing endocytosis or phagocytosis. It forms a vesicle known as a phagosome. The lysosome, created by the Golgi apparatus and filled with digestive enzymes, fuses with the phagosome, forming a phagolysosome, and the digestive enzymes act upon the bacteria, breaking it down. We now then have a residual body, a trash bag, if you will, that must undergo exocytosis to eliminate and secrete the remnants of the destroyed bacterium. We'll talk more about this in the immune system. The mighty mitochondria, a double membrane-bound organelle with the inner membrane folded, forming shelf-like structures known as cristae. And again, the mitochondria are involved in ATP production. The chloroplasts are somewhat similar in that they also are a double membrane-bound organelle that stacks of the thylakoids that contain chlorophyll involved in photosynthesis. Here's some plant cells, and you can see the chloroplasts within the cytoplasm of these plant cells. The centromere made up of the two centrioles, again made up of microtubules. In this case, we have 9 triplets and no central tubules. The two centrioles usually lay perpendicular to each other near the nucleus and will be involved in mitosis. I mentioned that the mitochondria and the chloroplasts are double membrane-bound organelles. The endosymbiotic theory, pioneered by Lynn Margulis, states that eukaryotic cells develop from larger bacterial cells that lost their cell walls and engulfed smaller bacteria in which they enslaved to make energy for the cell. Both mitochondria and chloroplasts resemble bacteria in size and shape, have their own DNA, can reproduce independently of their host cell, and contain 70 S ribosomes that are exactly the same as bacterial ribosomes. So, is it possible that at some point large prokaryotic cells decided to, one day, invaginate, form a nuclear membrane, and the membrane-bound organelles became much larger than bacteria and engulfed certain bacteria that were very good at making energy, and these became the mitochondria and the chloroplasts? It is possible because we know today that certain bacteria and protists actually have a symbiotic relationship where the bacterium is engulfed by the protists, and these photosynthetic bacteria make energy not only for themselves but for the protist, and the protist provides protection. Though they are still two unique organisms, just one inside the other, this is similar to what the endosymbiotic theory states. However, some people then try to push that a little further, saying, what are the fine extensions on this protozoan? Some people would say, well, these are just simply fimbriae from the bacteria that became the cilia. However, cilia and fimbriae have different protein structures, so I'm not sure the endosymbiotic theory holds at this point. Also, some people say, well, if the prokaryotic cell had flagella, the flagella simply became the eukaryotic flagella. But again, the basal bodies are different between the prokaryotic and eukaryotic cell. I want you to watch this video, and we'll discuss it actually in class.
A scientist and senior fellow at the Discovery Institute, Michael Behe is the author of the popular intelligent design book Darwin's Black Box and dozens of papers unrelated to intelligent design published in peer-reviewed science journals. Behe refused multiple invitations from NOVA to be interviewed for this program, though he went on record in the trial.
Dr. Behe, what is your profession?
I am a professor in the Department of Biological Sciences at Lehigh University in Bethlehem, Pennsylvania.
And you're a biochemist?
That's correct, yes.
How long have you taught at the college level?
For 23 years.
Sir, what is intelligent design?
Intelligent design is a scientific theory that proposes that some aspects of life are best explained as the result of design, and that the strong appearance of design in life is real and not just apparent.
Is intelligent design based on any religious beliefs or convictions?
No, it isn't.
What is it based on?
It is based entirely on observable, empirical, physical evidence from nature, plus logical inferences.
Now, when you use the term design, what do you mean?
Well, I discuss this in my book, Darwin's Black Box, and a short description of design is shown in this quotation from Chapter 9. Quote, "What is design? Design is simply the purposeful arrangement of parts. When we perceive that parts have been arranged to fulfill a purpose, that's when we infer design." End quote.
Part of the defense strategy would be to show the judge examples of biological systems they claimed were too complex to have evolved by natural selection, and therefore must have been the product of a designer.
Can you give us a biochemical example of design, Dr. Behe?
Yes, that's on the next slide. I think the most visually striking example of design is something called the bacterial flagellum. Now, this is a figure of a bacterial flagellum taken from a textbook, which is widely used in colleges and universities around the country. The bacterial flagellum is quite literally an outboard motor that bacteria use to swim. And in order to accomplish that function, it has a number of parts which are ordered to that effect. Now, this part here, which is labeled the filament, is actually the propeller of the bacterial flagellum. The motor is actually a rotary motor. And most people who see this and have the function explained to them quickly realize that these parts are ordered for a purpose and therefore bespeak design.
Under the microscope, bacteria powered by flagella seem almost acrobatic. They tumble, corkscrew, and pirouette, thanks to that whip-like filament. Driving this propeller is a tiny motor, part of a complex structure made of about 40 different kinds of proteins.
The bacterial flagellum looks like a sort of Jules Verne notion of what the future looks like. It has a strange sort of mechanical quality to it, these sort of cogs and waving tails and stuff.
And according to Behe, if any one of these parts is missing from the system, the motor can't function. Behe calls systems like this irreducibly complex, a term he coined. And he argues such systems could not have evolved naturally.
The idea is that there are certain aspects of life, perhaps organisms or organs or even cells, that in a sense could only have come about as a whole. In other words, it was very unlikely they could have come about through just a kind of contingent combination of parts over even millions or billions of years, but rather in a sense had to be created whole cloth all together at once, because everything fits together so well that to remove one part the thing wouldn't function.
Have other scientists acknowledged these design features of the flagellum?
Yes, they have. If you could advance the next slide. In 1998, a man named David de Rosier wrote an article in the journal Cell, which is a very prestigious scientific journal, entitled "The Turn of the Screw, the Bacterial Flagellar Motor." Now, David de Rosier is a professor of biology at Brandeis University in Massachusetts and has worked on the bacterial flagellar motor for most of his career. Now, in that article, he makes a statement, quote, "More so than other motors, the flagellum resembles a machine designed by a human," close quote. So David de Rosier also recognizes that the structure of the flagellum appears designed.
What I wrote was, this is a machine that looks like it was designed by a human, but that doesn't mean it was designed. That is the product of intelligent design. Indeed, this more has all the earmarks of something that arose by evolution.
Using an electron microscope, de Rosier produces ghostly pictures like this one, revealing the inner workings of what's been called the world's most efficient motor.
This is the drive shaft. This transmits this torque generated by the motor. That would then turn the propeller, which would push the bacterial cell through the fluid.
Michael Behe has argued that the flagellum could not have evolved, since its parts have no function for natural selection to act on until they are fully assembled. But evidence that refutes Behe's claim of irreducible complexity comes from a tiny syringe that injects poison, found in some of the nastiest of all bacteria.
This is a structure found, for example, in Yersinia pestis, the bacterium that causes the bubonic plague. And look at the similarities. Now, this structure doesn't rotate, but it still has to extend this structure, which is equivalent to the rod, the drive shaft here. It has to extend that because it needs this little channel. It's like sort of like a syringe. So the virulence factors that are made inside the cell, which is down here, can be exported, pushed up into this hole, and exported out through this long kind of needle, perhaps into a cell in your body or mine, and thereby create misery.
And it turns out the two structures look similar for a reason. The syringe on the right is made of a subset of the very same protein types found in the base of the flagellum on the left. Though the syringe is missing proteins found in the motor and therefore cannot produce rotary motion, it functions perfectly as an apparatus for transmitting disease.
So if we think about what it means to be irreducibly complex, the argument is that if you take away even one of these proteins, that the structure cannot function. And yet here is a structure that functions, that is missing several of the proteins, and yet here it is, a working, viable organelle of the bacterium. So indeed, this structure is not, in that sense, irreducibly complex.
To emphasize de Rosier's point, Miller arrived at court making an unusual fashion statement.
As an example of what irreducible complexity means, advocates of intelligent design like to point to a very common machine, the mousetrap. And the mousetrap is composed of 5 parts. It has a base plate, the catch, a spring, a little hammer that actually does the dirty work, and a bait holder. The mousetrap will not work if any one of these 5 parts are taken away. That's absolutely true. But remember the key notion of irreducible complexity, and that is that this whole machine is completely useless until all the parts are in place. Well, that turns out not to be true. And I'll give you an example. What I have right here is a mousetrap from which I've removed 2 of the 5 parts. I still have the base plate, the spring, and the hammer. Now, you can't catch any mice with this, so it's not a very good mousetrap. But it turns out that despite the missing parts, it makes a perfectly good, if somewhat inelegant, tie clip. And when we look at the favorite examples for irreducible complexity, and the bacterial flagellum is a perfect example, we find the molecular equivalent of my tie clip, which is we see parts of the machine missing 2, 3, 4, maybe even 20 parts, but still fulfilling a perfectly good purpose that could be favored by evolution. And that's why the irreducible complexity argument falls apart.
And this last slide I put in because I just really like birds of paradise. This was taken in Hawaii, so enjoy. Thanks.